Hello everyone, I'm Madison Bradley-Cronkwright, and during this video I'll be presenting the results of an investigation of cuboid elongation and variation in tarsal proportions of early primates. The ankle morphology of many extinct primates, along with other adaptations such as grasping feet and nails, suggests that early primates were likely arboreal. However, the specific locomotor niche of the last common ancestor of primates is largely unresolved. The notion that the primate last common ancestor was adapted for leaping locomotion was originally proposed in the late 70s and early 80s and has had a recent reemergence of support based on the elongation of postcranial elements such as the calcaneus in early members of our order, and because primates as a clade have greater calcaneal elongation than other mammals, where in this figure, non-primates are shown in blue, living primates are shown in black, and early primates are shown in red. However, it's also been argued that calcaneal elongation in primates is not exclusive to leaping specialists and is instead an adaptation that evolved to accommodate the loss of foot leverage that occurred in the acquisition of a mobile, grasping specialized helical metatarsal, which shifts the fulcrum of the distal limb segment from the metatarsal heads to the tarsus metatarsal joint or to the transverse tarsal joint. But what about the cuboid? While patterns of the calcaneus have been studied, the cuboid, which articulates with the calcaneus to form the midfoot segment, has been largely overlooked. There's been little inquiry into whether the cuboid contributes to midfoot elongation in early species, despite the cuboid's functional importance and its articulation with the calcaneus to form the midfoot segment. If and how the cuboid covaries with calcaneal elongation may also provide an additional source of information to assess the commitment of early primates to a locomotor repertoire that includes leaping. So ultimately during the study, I asked, does the cuboid elongate along with the calcaneus in primates, especially in primates near the LCA? And is the degree of cuboid elongation better predicted by calcaneal elongation, or is it better predicted by the size of the calcaneal cuboid facet, which is a feature strongly tied to body size? In pursuit of these questions, I took linear measurements from a sample of 10 genera of Eocene primates, including representatives from both the Adapiforms and Omomyiforms. For my comparative sample, I used 9 genera of extant strepsirrhines with various locomotor modes and body sizes, 4 genera of platyrrhines, and 4 genera of closely related outgroup mammals. All of the specimens used in my analyses are available on Morphosaurus. In order to investigate the relative proportions of the midfoot, first I calculated the degree of calcaneal elongation after Bohr et al. 2013, where you take the natural log of the portion of the calcaneus distal to the ectal facet divided by the entire length of the calcaneus. This is a functionally relevant measure because you're essentially measuring the relative length of the calcaneus that's distal to the load at the ankle. I then also measured the length of the cuboid and converted it into a proportion of the midfoot segment formed by the distal calcaneus and the cuboid. Lastly, I measured the area of the calcaneal cuboid facet since it has been shown to be a highly reliable predictor of body mass for both primates and the UR conta more broadly. It's also conveniently on almost all specimens, so it provides lots of good information to train models. To start, I ran bivariate linear models. First, I confirmed that within my sample, calcaneal elongation is negatively correlated with facet size, such that calcaneal elongation is negatively allometric. Primates, as a group, are offset from non-primate mammals by having distally elongated calcanei, with the largest residuals seeming to relate to locomotor mode. For example, a bush baby up here, or perhaps the Sloloris down here. When we look at cuboid length against facet area, again, primates are distinct from the outgroup mammals in having relatively long cuboids. When we add in the cuboid to get the full midfoot segment length, the relationship with facet area looks like this, where the offset between primates and outgroup mammals is more pronounced in larger species, and the relationship between midfoot length and facet area is more variable within the smaller bodied primates. Within the midfoot segment, the relative contribution of the cuboid to the total length is pretty consistent across all taxa in my sample. except for plesiodapus and the galligo, shown here in blue and pink respectively. What this ultimately looks like is the midfoot being comprised of around 50% distal calcaneus and 50% cuboid across taxa, except for the active small-bodied strepsirrhines like galligos or the chirogalids. And in this figure, you can see the variability in cuboid proportions in the strepsirrhines, especially the small-bodied chirogalids and the galligos. You can also see that despite their calcaneal elongation, Archicebus and Tihardina have midfoot proportions unlike other small-bodied primates. Instead, they have cuboids that contribute even more to their midfoot than is seen in most non-primates, with the only exception being plesiodapus. To get away from the constraints of looking at a series of bivariate linear regressions, especially since the relationship with facet area has the potential to mask other signals, I ran Bayesian multi-level models clustered by higher taxonomic groupings. Next, I'll show the results of one such model with the proportion of cuboid as the outcome predicted by the degree of calcaneal elongation, as well as facet area as a proxy for body size. When a model is run with the effective facet area controlled, almost all of the taxa included have a positive or weakly positive correlation between elongation of the distal calcaneus and the contribution of the cuboid to the midfoot segment, with the notable exception of extant strepsirrhines, which have a strongly negative relationship between the proportion cuboid and calcaneal elongation. The functional consequence of calcaneal elongation is an alteration of the mechanical advantage of the triceps surae, or the medial and lateral gastrocnemius and soleus. Increased distal calcaneal elongation means lower mechanical advantage about the ankle, or higher gearing. The higher gear of the ankles of taxa with distally elongated calcanei enables rapid plantar flexion, but it also requires greater input force per unit output force. Conversely, when the distal calcaneus is not elongated, the mechanical advantage of the ankle remains greater, and the ankle is able to be used in a low gear during plantar flexion. This lower gear allows for increased output torque per unit applied force, which is especially important for larger bodied animals since muscle force and mass scale allometrically. On the other hand, cuboid elongation, or lack thereof, doesn't actually impact the mechanical advantage of the triceps surae in tarsifocal mating primates, since any length it adds occurs beyond the fulcrum. Instead, cuboid elongation impacts the mechanical advantage of the deeper plantar flexors and digit flexors, such as the flexor hallucis longus. And I think that the reason that the cuboid elongation of larger-bodied strepsirrhines that are skilled vertical leapers is constrained is because strepsirrhines rely heavily on inverted and abducted foot postures to navigate vertical supports. In order to maintain forceful plantar flexion and digit flexion, which are both required for strongly gripping vertical and small diameter supports, the cuboid can't be too elongated and thus reduce mechanical advantage for the large-bodied leaping lemurs that are constrained by their available muscle force. In the future, I hope to expand my outgroup sample to add locomotor class into models, conduct ancestral state reconstruction, and include models with more rigorous phylogenetic control. I'd like to end this video by acknowledging these institutions as well as these funding sources. Thank you so much for listening.